Hello, I'm Jan Leifman.
Hi, I'm Gayatri from Menon.
Welcome to MedNews Week Conference, where we feature presentations by medicine's global leaders. Today we have an amazing keynote speaker in Dr. Adolfo Garcia-Sastre. Dr. Garcia-Sastre is the Director of Global Health and Emerging Pathogens Institute, Principal Investigator in the Center for Research on Influenza Pathogenesis and Transmission at the Tisch Cancer Institute at the Icahn School of Medicine at Mount Sinai.
In today's presentation, Dr. Garcia-Sastre discusses the latest developments in the treatment and management of COVID-19.
Did you know that Dr. Garcia-Sastre is a major contributor in the field of influenza viruses, who pioneered the development of reverse genetic techniques to allow recombinant influenza virus generation from plasmid DNA, the generation and evaluation of negative-stranded RNA virus vectors as potential vaccine candidates against infectious diseases like malaria and AIDS, the identification of the biological role of the non-structural protein of influenza virus during infection, the inhibition of interferon type 1 system, was also one of the initial members of the NIH Vaccine Study Section and Virology B Study Section, and a recipient of over 7 patents for his numerous contributions to the field of virology.
He's truly a global leader in the field of medicine.
So let's tune in and learn from this global pioneer.
So first of all, with therapies. Well, there are basically two types of therapies for COVID-19. There are the antivirals, which are drugs or biologicals that inhibit viral replication. And there are drugs also that they don't inhibit viral replication, but they are against severe symptoms. They are working by completely different mechanisms. An antiviral, something that stops viral replication, is making the best therapy when it's being given early on. Just after diagnosis, when the virus is starting to replicate, if you stop replication early on, then you are going to stop disease progression. And whether it's going to be mild disease or severe disease, by stopping the viral replication early on, you can stop the disease. However, when the virus has started to replicate already, and it has been several days, and people start to have severe disease, the ones that are unfortunate to have severe disease, then at the moment of starting symptoms of severe disease, the antivirals, they are less effective because the disease is not anymore caused by the virus that was replicating. The virus has stopped it, most of its replication is caused by an exacerbated response that happens in some percentage of infected individuals. And for that, you need a different type of therapy, and that's what the drugs against severe symptoms. These are the anti-inflammatories, like the dexamethasone, the IL-6 blocking antibodies, the anticoagulants that try to reduce the blood clots and the problems with coagulation that happens in people with severe disease. And these are good for people with severe disease. But in order to stop viral replication early on, the antivirals can be very effective. And for that, there are two types of antivirals. The ones that inhibit the viral factor, which are the ones that we know of that they have been in use for the treatment of COVID-19, including remdesivir, which is an inhibitor of the viral polymerase of the virus that is needed for the virus to replicate its genome, and it's an enzyme of the virus, and remdesivir inhibits this enzyme. It has the limitation that it needs to be used IV, intravenously, and therefore it cannot be suitable for taking easily, like an oral antiviral. Early on, we have also monoclonal antibodies, several ones. These are inhibiting the spike of the virus, binding to the receptor, and therefore the virus is starting infection. Again, it needs to be used intravenously, like remdesivir. And one of the limitations is that the virus is changing very quickly, and many of the monoclonal antibodies therapies that they were developed early on are not working anymore because the virus has changed And these antibodies, some of them at least, are unable to bind to the virus and to inhibit the virus. And that has become more apparent when Omicron came, which contained a lot of changes in the area that is targeted by these monoclonal antibodies. And we are lucky to have now oral inhibitors for the virus. This is Paxlovid and Molnupiravir. Paxlovid is an inhibitor of one of enzyme of the virus, the protease that is required for the virus to make its own proteins also. It has one of the problems that Paxlovid has is that it requires a booster. That's because the drug is being removed from the body very quickly. The booster is due to another drug, ritonavir, that allows to maintain the concentration of Paxlovid high enough in the people that take it. But ritonavir also is able to increase the concentrations of some other medications that people may be taking, and therefore has a lot of contraindications. Molnupiravir, which is an inhibitor of the polymerase of the virus, like remdesivir, unlike remdesivir, can be taken orally, but it's still unclear how many potential side effects they may have. I'm going to be talking about the antivirals that we have been working on, which they don't inhibit the viral factor. They actually inhibit a host factor, so one of our own proteins, and they inhibit one of our own proteins that is required for the replication of the virus. The viruses are very small entities. They require a lot of our own proteins for replication. And if we know of which proteins they are, the ones that they require, and if we find one of these proteins that can be inhibited without having toxicity, then we may have also a good antiviral against the virus, for which is more difficult to develop resistance, because the protein that is inhibited is not a viral protein. The virus can change very easily, but it's one of our own proteins, and our own proteins are not changing so quickly as the proteins of the virus. So the question we have early on when the pandemic came is, can we able to identify compounds that inhibit a host protein, the host factor that is involved in the SARS-CoV-2 replication among non-human drugs? And why among human drugs, and how this might be possible? First, if it's a non-human drug for which is already known, what are the side effects, if any, and what is how you can take this drug and what concentrations they can achieve in the body. This is information that is extra, so it's not like developing a completely new drug that you need to understand how well it's been taken, how much concentration it achieves, it's been easily degraded when you get into the body, does it have toxic effects? Non-human drugs, this information is already known. And why a non-human drug that is used for one indication may inhibit SARS-CoV-2 replication? It's a completely different virus that whatever a human drug is being used of. It has not been developed for this virus. Well, viruses use a lot of host factors, as I mentioned. So if it happens to be that one of the host factors that the virus is using is a host factor that is being targeted by a drug that makes an impact in some other disease, then we may repurpose this inhibitor to inhibit the replication of SARS-CoV-2. So how can we identify drugs that inhibit non-human host factors that may make an impact in SARS-CoV-2 replication? So for that, we have established a collaboration with Nevan Krogan and many people from a consortium that there is to try to understand what host factors are required for viral replication for different viruses, including SARS-CoV-2. They are located at the UCSF. And what my colleague Nevan Krogan did early on, as soon as it was known the sequence of the genome of SARS-CoV-2, is identify all the proteins that are encoded by the virus, which is a total of 29 different proteins. And then he expressed these proteins individually in human cells, and then determined by finding the host proteins that bind to these 29 viral proteins, what host proteins are interacting with these viral proteins. This is what is called the virus host interactome. And he identified a total of 332 protein-protein interactions. So what we postulate is that if a viral protein is interacting with a host protein, it has a good possibility that it does so trying to use this host factor for its own benefit, for viral replication. So we think that these 332 protein interactions is enriched on host factors that are required for viral replication. And now among these host factors is there any drug that is known that is being used for another indication that inhibits one of these host factors. And with that, there is 69 of these different factors that they are druggable, meaning that they have some drugs, 1 or 2 or 3, that are known to inhibit this particular host factor. So we took all these host factors. I'm going to show you the one that is more promising in our hands, and how we led to a particular inhibitor that is now in phase 3 clinical trials. So this is one of the factors, is called EEF1A. It's a host factor. This involves normally in protein translation, in making our own proteins. It was found to interact with 2 of the proteins of the virus, with the SARS-CoV-2. And it was known to have some inhibitors that have been used or tried to be used for the treatment of some cancers, Ternatin-4 and Pritidepsin. So we took these inhibitors, Ternatin-4 and Pritidepsin, and we tested for inhibition of viral replication first in tissue culture, outside of the body, in a cell line. The cell lines that we used in the beginning were cell lines where the virus replicates very well. These are the virus 6 cells. It's an African green monkey kidney cell line where the virus replicates very well. And then we look, is this compound, Ternatin or Pritidepsin, these compounds, able to inhibit viral replication? And we tested multiple things, and not only these ones, but some inhibitors of some of the host factors. But this was some of the most potent drugs that we found that was able to inhibit viral replication. And then how we look at that is we look for ability of the virus to replicate in cells in the presence of different concentrations of these particular inhibitors, either Ternatin-4 or Pritidepsin. And by that, we can determine what is the concentration that is required from this factor to inhibit 90% of viral replication, which is what's called the IC90. And Ternatin-4 was able to inhibit viral replication with an IC90 of 10 nanomolar. Now, inhibitors usually they are believed to be good when they're working at micromolar or submicromolar concentrations. So this is nanomolar concentrations, even 1,000 times less than when one tries to look for a good inhibitor. So that means that it's a very potent inhibitor. In addition, we look when it's able to have some cytotoxicity, and even though it looks like it's reducing cell viability in this broken cell, this doesn't it does not kill the cells, and this is this reduction in percentage of live cells, 50% that we see here, is not due to killing of the cells, but it's because the compound prevents the dividing of the cells that happens in tissue culture, and therefore you finish with less cells in the presence of the compound than the ones that you finish if there is no compound. And Ternatin-4 is working very well, but even better, Pritidepsin, which is also targeting the same factor, also called the other name that has Aplidin, and but it's the same compound, and the IC90 is in the nanomolar range, so even 10 times better than Ternatin-4. And the interesting thing is that Pritidepsin has been already approved for being used in some countries, specifically in Australia, for actually the treatment of multiple myeloma. And it's been used combined with dexamethasone. So that means that it does not have a contraindication with dexamethasone, which is one of the inhibitors that is being used in immunotherapy for COVID-19. Well, so once that we find that it has activity in viral cells, then we tried multiple other cell lines, not anymore these African green monkey cells, but also we tried human cells, human respiratory epithelial cells, like these derived pneumocytes, but some of the cells like 293s. Basically, there are different bunch of human cells. And what we find is that Aplidin or Pritidepsin has a very good inhibition activity again, at the nanomolar range for IC90s, which is even higher, meaning it requires less amount of the compound to inhibit 90% viral replication, than remdesivir, which is at this time was the only inhibitor that was available for this virus. Is working very well also even in primary cells, primary human pneumocytes, which are the main target of viral replication in vivo, and is working even after 4 hours after infection of the cells, while remdesivir start not to be able to inhibit anymore 4 hours after infection of a cell. So it looks like, at least in these tissue culture assays, plitidepsin is more potent than remdesivir. Now, what we also try to do is really plitidepsin working by inhibiting this factor, the EEF1A. And for that, we are lucky in that it's known what area in E1F1A plitidepsin is binding to and inhibiting this factor. And not only that, because tumors, they also mutate very frequently, like viruses. There have been some tumors that they have been treated with this inhibitor of tumor growth, plitidepsin, and they have developed resistance by a single point mutation in EYF1A that prevents binding of plitidepsin to this factor. This is the amino acid 399, a change of an alanine to a valine. Now, in order to demonstrate that plitidepsin is actually working in inhibiting COVID-19 SARS-CoV-2 replication by binding to EYF1A, we make now, through molecular engineering, cells in which we change this single amino acid in EYF1A, and we make them now this factor resistant to binding to plitidepsin. And when we do so, now plitidepsin here in red is unable to inhibit any more viral replication as compared to the original cells. If we use these cells that have mutated EYF1A for looking to the inhibition of remdesivir, this doesn't matter for remdesivir. It does not bind to this factor, so it doesn't matter to remdesivir. But this demonstrates that plitidepsin is inhibiting viral replication of SARS-CoV-2 by binding specifically to EYF1A, and is not by binding to some other factor that drugs usually bind to more than one factor, and sometimes it's difficult to know if they have off-target effects. So in this case, it's not an off-target effect, it's by binding to EYF1A, how plitidepsin is inhibiting viral replication. Now, knowing that, then we started to go into in vivo. For in vivo, we use animal models of SARS-CoV-2 replication and disease. One of the ones that has been more useful for that has been mice that are transgenic for human AC2. The reason why we need to use transgenic mice is because mouse AC2 is not a good receptor for SARS-CoV-2. SARS-CoV-2 binds the spike to enter the cells to human AC2, and human AC2 is required for entry. Mouse AC2 is a little bit different from human AC2, and the spike of the virus is unable to use this receptor. But if you make mice to express human AC2, these are these transgenic mice, now the virus will replicate in mice and replicates actually to high titers in multiple organs. So we took these mice, these transgenic mice that were developed actually not for SARS-CoV-2, but for SARS-CoV, the first SARS, because it used the same receptor, so they can be used also for SARS-CoV-2. And then we infected with SARS-CoV-2, and then we looked to viral lung titers at day 3. It's a very fast-growing virus in these animals. And we took some and leave it untreated, or treated with plitidepsin at a 0.3 mg per kg, 3 doses, or with remdesivir at 50 mg per kg, 3 doses, so a dose that is more than 10 times higher than plitidepsin. And when we do that, we get a comparable inhibition of viral replication, more than 2 logs inhibition of viral replication in the lungs, and also we don't get any more histopathology in the lungs. So we take these lungs, we look whether they look healthy. We have a score to show how sick these lungs are. And in the presence of plitidepsin, then we have almost no pathology of the virus causing in the lungs of this animal, indicating that it's working very well to prevent disease in mice. And with this data, PharmaMar, which is the company that actually developed plitidepsin as an inhibitor of myeloma. They have started clinical trials. This was the results from a phase 1, so a very small clinical trial where they looked mainly to toxicity that was found not to be toxic in the concentrations that were used. We used a concentration of plitidepsin that was based on the concentration that we knew that it was working in mice, and it has a similar behavior in humans with respect to concentration achieved in the lungs after taking plitidepsin. And there was only 50 patients per group, and not placebo, just 1.5, 2, and 2.5 mg per day for 3 days of these people. So again, no side effects. And that was at patients that they were hospitalized at the moment of hospitalization. And what we found is that at the higher dose, you can see that there is less days that the patients stay in the hospital as compared to the lower doses. And there is also a more quickly removal of symptoms from the people that from this very small trial. So there is a tendency here to a beneficial effect of treatment with plitidepsin. That's not completely proven because it's a small trial, there is no placebo, but that indicates no severe events and a tendency with a higher concentration to get the benefit. And with this now, there is a phase 3 trial going on, with now there is more participants, it's 109, and in multiple hospitals, where now there is also placebo arm, so people are just treated with the standard of care, whatever is available in these hospitals. Many of the standard of care treatments is with dexamethasone for people that are being hospitalized, and with different amounts of plitidepsin in order to find out whether actually these 2 dose levels of plitidepsin versus control is reducing the number of days in the hospital of people with severe COVID-19. And we hopefully expect this trial to be finished by December 2022. So if that works well, then we'll have one more drug that could be used for the treatment of COVID-19 that could be added to the already existing drugs. And perhaps this could be beneficial because of, you never know how many drugs you need for this virus and whether the viruses are going to acquire resistance or not to the drugs that are already used. All right, then I'm going to change to vaccines. We have also been working on vaccines, and vaccines has been one of the highest success of the story of the pandemic of COVID-19. Perhaps it doesn't look like, because COVID-19 still is there, because there is still a lot of SARS-CoV-2 infection, but it's becoming clear that the percentage of severe disease that the virus is inducing in vaccinated people is very low as compared to the percentage of disease that you get in non-vaccinated people. So even though the vaccines are not able to completely prevent infection in most of the cases, due to a combination of, with the time you get less immunity, also the virus has been changed, evading immunity, and now there is still a lot of infections in vaccinated people. In general, these infections are not as giving severe symptoms as compared to infections in non-vaccinated people. So that's the success of the vaccines. But certainly, because there is a lot of infections, we think that there is room for improvement. And actually, the vaccines that we have not been able to be deployed in all parts of the world due to the limitations with respect to cold chain distribution and availability of these vaccines in different parts of the world. Now, there have been many strategies that have been used for vaccines, and the ones that they have been in use right now in the United States have been the RNA vaccines, both the Moderna and the Pfizer-BioNTech vaccine, and the viral vector vaccines. The one that has been more used is AstraZeneca, based on adenovirus vector, but also Johnson & Johnson had the single dose, also based on adenovirus vector, that has been also being used for reducing severe disease after infection with SARS-CoV-2. The one that we have approached is been using also viral vector, but this is not an adenovirus vector. It's a vector from another virus, which is a virus that comes from chicken. It's a chicken virus, it's an avian virus, The name of the virus is Newcastle disease virus. Most of the people are not familiar with this virus because it's not a human virus, but it's a big problem, Newcastle disease in chicken. And it was recognized that that was a big problem in chicken farms since the 50s. And this led to development for a vaccine in chicken against Newcastle disease, which is based on a strain of Newcastle disease virus that is attenuated, that does not cause disease in the chickens, but induce immunity against Newcastle disease viruses that are able to cause disease in chickens. And because of that, when these vaccines were developed, they started to vaccinate chickens all over the world, and it's still the case. There are billions and billions of chickens that have been vaccinated every year against Newcastle disease based on a virus that is an attenuated Newcastle disease virus that is being grown to high amounts because you need to vaccinate a lot of chickens. And that is also very cheap because you cannot afford expensive vaccines for chickens, since if not, the price of poultry and eggs will be too high. So we thought, based on this, that it's very easy to produce, and you can actually make billions and billions of doses, that it could be an ideal vector for expressing an antigen from SARS-CoV-2, in this case a spike, and see whether it's able to induce an immune response once that you do that in humans, not anymore in avians, in humans against SARS-CoV-2. And that's something that we have been developing together in Mount Sinai, together with my colleagues Peter Palese, Florian Kramer, and Willa San. So what potential advantages this virus could have versus existing approaches? Well, with respect to the adenovirus vectors, the Newcastle disease virus vectors, they have no pre-existing immunity at all in humans, because we are talking about an avian virus, not even a human virus. One of the main advantages that they are being manufactured using the same technology, which is being grown in embryonic eggs, as influenza virus vaccines. And what does it mean? That almost every country has a factory for making influenza virus vaccines, so they can deploy these factories to make now a vaccine based on Newcastle disease virus. The virus and the virus vector is stable at 4 degrees, so it has less problems with cold chain as compared to the RNA vaccines. And then, is it safe? Well, fortunately, there have been several clinical trials that have been done with Newcastle disease virus as potential vaccine for cancer. And in these trials, we have learned that this virus is safe and does not induce adverse events when administered in people. So how we make a vaccine? This virus is an RNA virus that has a single RNA segment that encodes for a total of 6 viral proteins. And then what we do is, by genetic engineering, we introduce one more gene into the genome of the virus and make it express, in addition to the 6 viral proteins, the spike of SARS-CoV-2. But we have a modified spike. We make it more stable by eliminating a furin cleavage site that is required for activation of this protein during entry of the SARS-CoV-2, and we insert some prolines in the area of the spike that makes this protein also more stable. So we have a stabilized spike that is better for inducing of antibodies. And in addition, now we include in the protein, instead of the normal transmembrane cytoplasmic tail, where this protein gets anchored in the SARS-CoV-2 virions, we put the cytoplasmic and transmembrane domains of one of the proteins of Newcastle disease virus. So what this results is a Newcastle disease virus that now, in addition to have its own glycoproteins in the envelope of the virus, also has the SARS-CoV-2 spike in the envelope of the virus. It's a protein, again, stable, and is not functional, cannot use it for entering the cells because of the mutations that were introduced in this protein. And it expresses very well when we look at when we have purified virus and we look to the protein content of these viruses. Then we looked at this compared to the wild-type Newcastle disease virus. This one that expressed this hexaproline spike, that's how we call it, XSP spike, contains a very prominent band here, the band corresponding to the spike. So it expresses a nice amount of a spike in infected cells and also incorporated into the virion particle. And this allows us to be used in 2 modalities, and that's what we are trying to test now in clinical trials. Again, we grow these viruses easily in embryoid eggs. The same substrate is used for growing influenza virus vaccines. We harvest these eggs, and then they contain high levels of this virus vector, virus vaccine vector, and we can either inactivate it, the same thing that is done with the influenza virus vaccines, and then because it has the protein in the virus, then we can use it directly as vaccine by the intramuscular route, systemic route, the same as it's been done for the other vaccines that exist right now for SARS-CoV-2. But we can also use it live, because it's a virus that does not induce disease in humans, and now use an intranasal route. And this may have the advantage that now you induce an immunity against the spike, not only through circulating systemic antibodies in blood that protect very well from replication in the lung and from disease, but also you induce an immune response in the upper respiratory tract. That is the place of infection. So perhaps an intranasal vaccine will stop more infections than an intramuscular vaccine, which are the ones that we have right now. So how are we doing? We have done several preclinical studies first in 2 animal models, mice, again, that's expressing the human ACE2, and golden Syrian hamsters, which is a good animal model for replication of SARS-CoV-2, where the virus replicates without the need of introducing the human receptor. It's able to use the receptor of hamsters. And we have tried 3 vaccination regimens, 2 doses, starting with inactivated vaccine, intramuscular and intramuscular, a live vaccine, intranasal and intranasal, and live vaccine, which we start with intranasal and we follow with intramuscular. And what we have learned from these preclinical studies, what we have learned is that it's safe, does not induce disease in these animals, is immunogenic, induces high levels of antibodies, and is protective against challenge with SARS-CoV-2 in these animals. And this is one of our first publications with that came out using this virus in an animal model, this vaccine. I'm just going to show a few data in animal models. This is one in hamsters, in which we have used a modality, this intranasal live, given twice at day 0 and day 22, prime boost approach. We take blood for looking for antibodies, and then we challenge at day 44 with SARS-CoV-2 virus these animals. Now we know that we induce nice levels of antibodies against SARS-CoV-2. Even it looks like one single dose is enough for getting good antibodies. These antibodies not only neutralize the wild-type virus, but also neutralize different variants. This is the beta variant, and this is the alpha variant. And when we challenge with SARS-CoV-2, the control animals, they lose weight, they have disease, but the vaccinated animals, they don't lose weight, they don't have disease. If we look to replication of SARS-CoV-2 in the challenged animals in the lungs of these hamsters, the control ones, you get high levels of replication of the virus in the lung, but you get complete protection against replication with this against SARS-CoV-2 virus, but not only in the lung, but also in the nose, which is the upper respiratory tract related to transmission. We get this very good reduction of our replication in the nose. So we think that if that's the same thing when we vaccinate people, then we'll have a good protection not only against disease, but also against infection and transmission. Now, based on that, we have started clinical trials, and we have been fortunate enough to collaborate with different governments from different countries, as well as here in the United States with money from different philanthropy in order to move forward into clinical trials. So right now, there are 5 ongoing clinical trials with the Newcastle disease virus expressing a spike. 3 of them, they have a inactivated modality conducted with the help of the governments of Thailand, Vietnam, and Brazil. In Thailand, in Thailand, there's been given intramuscularly, inactivated, with and without an adjuvant. There have been a phase 1 showing a very good lack of adverse events and even less reactogenicity than the RNA vaccines. And phase 2 has now been also finished. The data, I would show a few of these data. They saw very good immunogenicity, very good induction of neutralizing antibodies. Phase 3 now, which will be the last step to see whether this vaccine is working. The protocol has been submitted, and we have the commitment of the Thailand government to conduct the phase 3 study. In Vietnam, it's very similar, phase 1, phase 2 with an inactivated vaccine. And the phase 3 protocol has been submitted. In Brazil, we come with the Institute Butantan, where they have done already phase 1, and the phase 2 and 3 is in planning phase, but it looks like they are also committed to continue. And this again, all of these 3 is intramuscularly inactivated. Now in Mexico, we are also collaborating with them, with the government of Mexico for these trials. We have basically given the vaccine seed to these countries, and they have grown the vaccine in their own facilities, and they have been conducting their own clinical trials. So in Mexico, it's a live modality, the one that we are using. We have conducted already phase 1 trial, intranasal, with we saw good immunogenicity and again, no adverse events. And there is a phase 2 study now going on, which is a booster study, where it's been looking to people that have received 2 doses of vaccination, either RNA vaccines or adeno vaccines, or inactivated vaccines that have been using also in Mexico, and then see how well it boosts. And then we have started also clinical trials using also intranasal vaccine in the United States, in Mount Sinai, where we have an ongoing phase 1 trial to see how good in the boosting assay, again, people that have been already vaccinated, how good antibodies is able to induce, especially intranasally, and if these antibodies are able to prevent infection. So I'm just going to show a summary of some of the data in terms of immunogenicity. This is the trial that has been conducted in Thailand with different doses of the vaccine with or without adjuvant, 1 microgram, 3 micrograms, and 10 micrograms. And these are the levels of neutralizing antibodies that one obtained in these individuals, with 1 dot being 1 individual. And we get high level of neutralizing antibodies, comparable or even better than the neutralizing antibodies that one gets with the RNA vaccines. This is the Pfizer vaccine, and higher than the antibodies that you get with natural infection. So it looks quite good. And these are the results now in the different country. In Vietnam, for phase 2, where 3 micrograms and 6 micrograms, in this case the vaccine was called Covivac, but this is the inactivating Newcastle disease virus expressing the spike with the stabilizing mutations. They have been compared with the AstraZeneca vaccine. And the interesting thing is that we get higher levels of antibodies in the people that receive the Newcastle disease virus as compared to the people that receive the AstraZeneca vaccine. So it seems that it's working quite well in terms of induction of neutralizing antibodies. So hopefully we can continue with the phase 3 clinical trials and see whether we can develop this vaccine that again has several advantages from the previous one, especially with respect to easy manufacturing and easy administration. So in summary, I saw you first in terms of therapeutics that SARS-CoV-2 interactome provides with rational therapeutic targets for drug repurposing, that plitidepsin, an inhibitor of EYF2A, has potent SARS-CoV-2 antiviral activity both in vitro and in vivo, and we are being conducting now, or they are being conducting right now, phase 3 clinical trials with this drug. And then we have developed a vaccine that is still in clinical development, based on Newcastle disease virus expressing the X6 protein spike that is stabilized, that is highly immunogenic, safe, and induce high levels of neutralizing antibodies, and hopefully mucosal immunity when we administer intranasally. At least that's what we know from the hamster studies, and therefore it can make also a higher impact in transmission. And with that, this is the different agencies that have been helping us to conduct these studies, thanks to the funding that we have received from NIAID and DOD and DARPA and multiple philanthropy foundations, as well as the governments of Vietnam, Thailand, Brazil, and Mexico for the vaccine. And these are some of the people that have been working on in my lab during this time in SARS-CoV-2. And I need to thank them because they are the actual people that have been doing the actual studies, as well as the patients that have been volunteered for the clinical trials.